limited. Two structural facts bear on how the committee should weigh that. Number one, KPV is a 3-amino acid fragment of alpha MSH, an endogenous human peptide. And number two, as a tripeptide, KPV carries a lower aggregation and immunogenicity risk profile. The most probable source of patient harm is product quality, misidentification, impurities, and contamination. That is precisely what our conditions target through Drug Master File sourcing, defined impurity limits, and independent lot testing. We support the approved topical and systemic therapies that exist today for wounds, psoriasis, and eczema, and they serve many patients well. But inflammatory skin and wound disease is heterogeneous, and a meaningful population fails or cannot tolerate corticosteroids, calcineurin inhibitors, and biologics. For those patients, a compounded anti-inflammatory tripeptide gives prescribers an additional individualized option. And as with the other substances, the alternative to a conditioned pathway is not the absence of KPV. It is the current market where KPV is already sold without identity testing or oversight, posing risk to that population. We add that KPV's small size makes it comparably tractable to characterize and test, which strengthens the case that a conditions-based listing can be implemented reliably for this substance. In addition, our comment includes proposed safeguards that should be included as mandatory conditions when adding these peptides to the bulks list. In brief, we propose that compounders only use API sourced from manufacturers that maintain up-to-date Drug Master files and that have had a satisfactory FDA inspection. 2, each bulk peptide lot should undergo independent third-party testing for critical quality attributes. 3, sterile peptide products should be subject to enhanced sterility and quality controls. 4, compounded products should identify both the bulk ingredient manufacturer and compounding pharmacy. And lastly, compounders should maintain complete lot-level chain of custody records from manufacturer all the way through dispensing. Thank you.
Thank you, Dr. Baker. Speaker number 8, please. Please state your name, any affiliations, and you have 3 minutes.
Good afternoon. My name is Joshua Sweeter, Summit Works USA, a domestic API manufacturer in Naperville, Illinois. My background, again, is an analytical chemist. I speak today on the characterization and controls behind it in the first criteria. KPV is just a tripeptide with 3 amino acids only, a short, fully defined molecule that's among the most straightforward to characterize and identify, which makes the data from the market study we just conducted very striking. Of our blinded quality control test of the R&D, the research only, market found that KPV was the worst performer of any substance we examined. Of 7 products, not one came within 10% label claim. Every one was overpotent, from up to 80% overpotent. From them, we looked at chromographic purity. One of the samples had 30% unknown product in it, so 70% pure with 30% unknown. Here's where the failures should have been controlled. Potency. Quantitative HPLC assay against a reference standard with a release specification. Every one of these market products would have been rejected and never shipped if in the compounding market. Purity, chromatographic purity with defined totals and single purity limits. So a 70% pure lot would have never left the building. Identity, high resolution mass spectroscopy plus retention time on every single lot would have confirmed the identity easily. Residual TFA counter ion is a clear example of what testing catches. It was talked about before, but of these 7 products, 3 of which contain high amounts of TFA. One carried nearly 20 milligrams per gram. That's 2% TFA. I don't think anyone in this room would agree injecting 2% TFA would be a safe thing to do. We quantified TFA in every single lot as a defined limit alongside a full panel of residual solvents, micro elemental impurities, and water content. All of these are third-party verified at a domestic facility built to CGMP standards. Characterization is the first criteria. Safety, effectiveness, and historical use are separate and outside my scope of work. What these controls settles is identity, dose, purity, and is exactly what the current market fails, but the compounding market could control. KPV is sold to U.S. customers today online as research use only, outside FDA oversight, and our data shows it failing accuracy every single time. Domestic manufacturing under U.S. oversight with these release controls replaces the current problem. We support adding KPV to the list. Thank you for your time.
Thank you very much. Speaker number 9, please. Please state your name for the record, any affiliations, and you have 3 minutes.
Hi, my name is Dr. Matt Cook. I'm a physician and I work in a big group medical practice, and we've been doing peptides for 10 years. And we estimate that we've done probably at least 15,000 patients over the last 10 years actively treating patients. I'm a co-founder of Vesalius Longevity Labs and co-founder of an AI platform that's going to track labs, genetics, and wearables, and an IRB-approved database that will give us some real-world outcomes to help us answer some of these questions. KPV is probably the easiest to understand of all of these peptides. It's only 3 amino acids. And because of that, it has significantly less immunogenicity, and it really falls below both from a T-cell epitope and from an antibody-mediated mechanism, immunogenicity compared to bigger peptides. It also has several mechanisms of having an anti-inflammatory effect. And it's something that we've used quite a bit in clinical practice. Go ahead and go to the next slide, please. And so then, the big use where we've had a benefit is in gastrointestinal illness, in irritable bowel syndrome, and inflammatory bowel disease. And inflammatory bowel disease, this has been in patients who are on maximal medical therapy, but they're still continuing to have symptoms. And we've seen benefits on those. We've also seen benefit on people who are having side effects of their medical therapy, and so they use it as an adjunctive therapy. And then in many of the patients with gastrointestinal illness, they're combining KPV and BPC. In general, gastrointestinal problems will relate to general inflammatory conditions. And so we've seen benefits in that category as well as in the setting of skin. What I'll tell you is that this is one of the most vulnerable populations that I've seen in my life, and they tend to react to almost everything. And I've basically never seen anybody have a problem from KPV in many, many hundreds of cases. The other category that we use KPV for a lot is in patients with histamine-related conditions, and especially mast cell activation. And that was a condition that didn't even really exist when I went to medical school 30 years ago. But it's something that I think has really come up in the this era of post-COVID. And so we've seen people use less medications to treat their mast cell activation, and have had a very good experience with that. And so overall, what I'll say is that in our hands, this has been a very safe peptide, and I encourage you to approve it today. Thank you for your time.
Thank you. Okay, speaker number 10, please. Please state your name, any affiliations, and you have 3 minutes.
Hello, my name is Dr. Alex Tatum, and as I mentioned previously, I'm in private practice urology in Indianapolis. KPV is a tiny peptide consisting of just 3 amino acids, lysine, proline, and valine. It represents the working end of alpha MSH, a hormone that your body already uses to switch off inflammation. It has been studied for 5 decades by independent academic groups spanning 3 continents. Next slide. And over that time, a consistent picture has emerged. KPV calms inflammation at the cellular level, shutting down the NFKB switch at just nanomolar concentrations with anti-inflammatory activity comparable comparable to large corticosteroid doses in mouse models. Across 3 separate mouse models of colitis run by different labs, KPV calmed the inflammation that drives tissue loss and cancer, and it reaches those tissues through an elegant mechanism. The inflamed colon overexpresses the PEP-T1 transporter that pulls KPV into cells, so the molecule concentrates right where the disease is. In a corneal wound healing model, every KPV-treated eye was fully healed by 60 hours, while none of the placebo eyes were. Next slide. Now, a fair question about any anti-inflammatory is whether it weakens the immune system. KPV doesn't. In laboratory studies, rather than lowering host defenses the way corticosteroids can, KPV killed bacteria directly, including MRSA, and it enhanced rather than impaired the immune system's own killing of pathogens. That distinction matters. It calms inflammatory signaling without simply turning the immune system off. Next slide. Its close cousin, KDPT, went into a randomized double-blind phase 2A trial in ulcerative colitis. Remission was significantly higher at weeks 2 and 4, and all doses were reported to be well tolerated. KDPT is not exactly identical to KPV, and there is no phase 3 on KPV itself. But under the compounding standard that Congress wrote, that is not the question. That question is one of safety. And in the best documented pharmacy network from AAPM Partners, KPV was dispensed in over 23,000 vials, and the complaint count was 0. I must note that was submitted before the July 9th date to the FDA with assurances that that information would be provided to the committee. My understanding is that it has not been. Passive data may not be a clinical trial, but 0 complaints across documented use is evidence this committee should not ignore. Next slide. And here is the problem with the COA that FDA uses for KPV freebase. The face of the document says UK peptides, research only. The seller's own page says the product is not validated for medical applications. A research-only certificate cannot fairly represent the qualified bulk API sourcing and pharmacy release records behind a legit 503A script. The FDA's own evidence proves why patients need a regulated medical pathway, not a closed door that forces them into the gray and black markets. Next slide. KPV was pulled into category 2 at the same 2023 action with BPC. FDA's own 2026 search found no FAERS report and no published human adverse case event for KPV. I ask that you please recommend KPV freebase and acetate for the 503A bulks list, restore it to category 1 while rulemaking proceeds, and make it available to patients and physicians once more. Thank you very much.
Thank you, Dr. Tatum. Okay, speaker 11, please state your name, any affiliations, and you have 3 minutes.
Hi, my name is Lee Rosebush. I'm chair of the American Academy of Peptide Medicine, OFA, as well as an attorney for multiple compounding pharmacies. Secretary Kenny said it best. FDA and HHS's job is not to recommend a product, or in this case, approve a product, but it's to provide access to a product. Yet during the review of these peptides, including today, the FDA has been pushing for the FDA new drug approval standards to apply to the consideration for inclusion of these peptides on the 503A bulks list. I want to be abundantly clear. That is an improper and illegal standard that's being applied to this consideration. In fact, FDA has developed its own regulation under 21 CFR 216.23, which we went through earlier, which is up on the screen now, that regulates the standard that should apply for the consideration of a substance on the bulk substance list. And these standards do not require any human study or proof in any way. Like with BPC, AAPM has provided additional data that has not been included in the packet that was provided to you. For example, AAPM conducted a retrospective analysis, one of the largest to ever occur in the peptide space, of over 3.6 million doses of KPV. That data was provided in our exhibit. The adverse event rate was literally less than 1/100%, and if you look at adverse events that were serious, it was literally 0. As noted on the screen above, there's no requirement to prove safety or efficacy. Accordingly, the proper standards are physical, chemical characteristics of the API. Notice it doesn't require proof that the API can be safely made or is currently available. Instead, you just need to provide characteristics of the substance. Next. We went through the data last time with being withdrawn. As you can see in this perspective, AAPM has provided this material. You can see the molecular weights, the molecular formulas. We have provided US names. Next. Mass spec weights. Next slide. You can see the aspects that we went through here. We partnered, just so folks know, with ARL, Tom Kupik specifically, to be able to do this. We actually ran it through mass spec, took it down from mass spec down to its relevant salt in this case. We have both acetate weights in our thing as well as base weights. You can compare for dosing perspectives. By the way, we did that for BPC, even though it was mentioned last time. All of that is in your record. We used USP standards. Next slide. You can see here the USP 795 and 797 for the testing aspects of this, as well as for those that are registered for 510. Go ahead, next slide. You can see the standards that we used. All of these aspects have been included. 467, 1503 is an example for both USPs. Next slide. So in essence, we've created, next slide, we've created, from that perspective, a USB light monograph. Here's what was used for others, as you can see as an example for the ones that have been pulled. Next slide. These are old ones. Next slide. There's the KPVs. Next slide. Here's what we provided from our record from that perspective, including all the analysis side of things, immunogenicity aspects discussions, impurity discussions, including what that would look like. We want to highlight that FDA's briefing documents include materials from previous nominations to FDA that have been withdrawn, as I mentioned before. Furthermore, AAPM understands that establishing accurate peptide content via absolute analytical methods is critical for qualifying in-house reference standards. In the absence of a USP reference standard for calibration using an HPLC assay, the absolute peptide content may be corrected by subtracting the total related impurities determined by HPLC, which we have done. We've provided these reference standards in our submissions. We are giving them away for free. So there's no doubt on the characteristic aspects of this. The 3.6 million doses from that perspective gives you the safety side. And you just heard from that perspective, that includes for historical use, as well as the efficacy discussion that Alex just had and the additional materials that we have provided. Accordingly, all 4 criteria are met, and this one should be added to the 503A bulks list as well. For the record, that also includes the weights from that perspective if you were to use the acetate and transfer to a base and vice versa. All of that is included inside of our docket.
Thank you, Dr. Rosbush. Much appreciated. Okay, speaker 12, please. Please state your name, any affiliations, and you have 3 minutes.
Certainly. Good afternoon. My name is Honey Sagari, and I'm founder and CEO of LEMD, a national telehealth platform, as well as a lifelong student of aging and disease and an advocate for the hundreds of thousands of patients and the physicians who care for them. Today, I'm here representing a community that is predominantly women. For many of these women, this discussion is about far more than peptides. It's about finally feeling heard after years of living in chronic inflammatory conditions that have affected their health, their families, and their quality of life. They have entrusted me to advocate for one thing: safe access under physician supervision. My academic background includes an advanced studies in biochemistry and gerontology. Earlier in my career, I built and led a home healthcare company serving patients with Alzheimer's and dementia. Using research-informed approaches beyond the conventional standard of care, we cared for many of these patients who maintained their independence and quality. Watching families gain more meaningful time with the people they love changed the way I think about medicine. It taught me that when patients consistently do better than expected, those signals deserve scientific investigation, not dismissal, and that's how I feel about KPV. I've read the agency's reviews, and I'm not here to argue with it. The concerns regarding characterization, manufacturing consistency, impurity profiles, and product quality are real, and I'm not asking you to overlook them. I would simply ask you to recognize that those concerns describe. They describe an unregulated marketplace, not a regulated 503A compounding framework. Those are exactly the kinds of problems physician oversight, pharmacy standards, and rigorous quality testing are designed to solve. Patients are already seeking KPV today. They're using it. That's not the question. The only question is where they obtain it. If the regulated pathway disappears, many of these patients will not stop searching. They will turn to research-grade products sold online with no physician, no pharmacist, no patient screening, no quality control, and no meaningful accountability when something goes wrong. Closing the regulated pathway will not close that market.